be disorder from birth, or you cross certain threshold, it is actually a biochemical problem. And again, like addiction, it's a food addiction, and you need to treat it chemically, biochemically, okay? And then lastly, the world's population is aging, and there are age-related problems, and how we can address that, whether it's Alzheimer or whether it is malfunction of other organs and whatever, we need to treat that. So these are healthcare challenges that nanomedicine provides solution for. On my book on nanomedicine, I have touched on many of these, talked about how nanomedicine can provide possible solutions. Some of them can be realized in shorter term, some of them in longer term. So one first part I'm going to talk about is example of this is light. How one can utilize light for diagnostics and therapy and incorporate that in nanoparticle formulation to advance nanomedicine. And it's not surprising that light is very important, has played a very important role. Light is credited for, in a way, evolution of life, you know, and it involves some very simple things like photosynthesis in the plant that is important for our survival, to other kinds of effects. Light has been used in Indian mythology for treating certain kind of skin diseases and others. So light is very important, and it also controls our neuronal response. You know, there is a light out there, we feel great. When it is dark and gloomy, you don't feel so great. So we are utilizing light as also fundamental tool, and already light is in healthcare diagnostics. Okay, so for example, you can see this is digital finger oximetry. If you go to the hospital and they bring and they clip your finger to measure your oxygen level in your blood, okay, and it's just looking at the ratio of concentration of oxy to deoxyhemoglobin, oxygenated, because their absorption profile is shifted. So here is again an example of how we can utilize light and physics, okay, a light matter interaction to develop very powerful, very simple device that just takes laser light going through your finger and look at the absorption of oxyhemoglobin, which is shifted, versus deoxyhemoglobin, and that ratio gives you oxygenation. Very simple. Okay. Second one is colorimetric test, where you could have a strip that actually has antibody immobilized, and then one can put a specimen of urine, and the urine can have signature of pregnancy. It has antigens that binds there and changes the color of the strip that has certain kind of nanomaterials, okay? And when it changes, in this case it's metal, nanoparticles, its color changes. And with the color change you can detect at home whether the woman is pregnant or not pregnant. And this is in market, okay? The other one is flow cytometry, where you take a cell suspension and we flow through channel and interrogate what kind of cell population you have, which cells is called flow cytometry. It's a very powerful clinical tool. So these are some of the examples of light. Then you also know the laser surgery has played a very important role, particularly in area of ophthalmic, right? Eye vision correction is very important one. You can go and you can do laser surgery to correct for your vision. Or you can, there are other kinds of retinal detachment, you know, macular degeneration, many kinds of treatment light can do. So light is in marketplace, and what we are trying to do is expand it biophotonics. So this is the outline of the course where the first lecture, set of lectures, is part 1. Doesn't have to be covered all today. Okay, we will talk about using light as a tool that will advance nanomedicine, so biophotonics. We will introduce some concepts. We can talk about some optical imaging techniques in medical, and then we talk about sensing and diagnostics. Then we will introduce nano to that, nanobioengineering in part 2, how we can design, make nanoprobes through chemistry, how we can enhance imaging and sensing, and what kind of nanoparticles we can use with certain kind of functionalities that we can incorporate. Then we will talk about nanomedicine, which is light activated therapies, combining that with imaging diagnostic, that's called theranostic. And then we talk about some of the toxicity concern and biocompatibility, and what kind of tests we can do to test them, feel that they are safe. And then we will, last part, we will talk about what are the challenges to take it actually towards patient care, okay? And what are the opportunities for many PhD theses in different areas, and many technology that can be advanced to actually benefit society, okay? All right, so let's move on. So the first part, realizing that many of you are not biologists, many of you are chemists, and some are physicists. Those of you who are biotechnology, you know this material already. So luckily for you, I only have 6 basic slides, okay, so you wouldn't be too bored. But maybe perhaps the chemist physicist can be enlightened a little bit. And I just want to tell you a little bit over there that I never took a formal course in biology, okay? So biology, they were intimidating to me, but learned slowly and slowly as I needed them. So then the lesson I want to, or message I want to give to you who are not biologists, don't be intimidated. They do appear very complex, and they are, because they involve many chemicals and many different structural levels, and they all kind of are coupled together. Our body is a system coupled together, okay? So it's very complex, but you need to dissect from very basic fundamental level. So I wrote this chapter, Fundamentals of Biology, in my book on biophotonics, only for the people who don't know much of biology and are scared to open a thick book. Mineral, and all these chemicals are lifeless. You prepare them in chemistry lab, but they're not living. They don't replicate. They don't repair. But nature assembles them into units that can replicate, that can be living. This is one of the fundamental challenges now of how we can combine them to make them living. And tissue engineering is one area that promises to do that, some ways. So their system builds in, so they combine together to form macromolecules, DNA and RNA, proteins and lipids, the combined fatty acids, carbohydrates, okay? And then you can assemble them in the basic fundamental building block of life. These are cells, right? And so you can have different kind of organisms. One is virus, and viruses are very small, 20 to 200 nanometer size. So they are truly nano objects of nature, right? And they just contain single or double strand. Viruses mutate, and this like HIV virus, people even call it a smart virus, that it mutate around this, very difficult to form any vaccine and others to control them, and yet they are non-living, okay? Then you have these prokaryotic cells. These are cells of bacteria. So at a higher level, the bacteria are there. They could be from 500-600 nanometers to micron sizes, and so they can be much bigger than viruses. So you can say them they are submicron to micron or nano to micron, okay? And these are structured single cell consisting of a double closed compartment, but still lacks a nucleus. There is no central nucleus in there in this bacteria. They are called prokaryotic cells. And then they are also replicating, okay? They can replicate, and they can survive outside too. Okay, so they are definitely living. Okay, then you have these eukaryotic cells, which are anywhere from 6 nanometers to 25 micrometers to 25 micrometers, much larger. Okay, so and our body, or most or the animals that we encounter, they all consist of this kind of cell. Okay, and here the big difference from here is that you have a nucleus. You have a big part inside the cell is a nucleus. So cell has a structure, and those structures have different compartments. These are called subcellular compartments, and the other name for that is organelle. Organelle is organs of a cell, small parts, right? A nucleus is an organelle. Mitochondria, where the energy is produced inside the cell, is an organelle. Okay? So the difference between these two, as I said, one is a nucleus, the other one is a membrane structure, and they are self-replicating, self-healing, and also assembled together to form a bigger organism, which is called tissues. Okay? So again, if you want to look at under microscope, ordinary optical microscope, you cannot see the viruses. They're too small, because your resolution, most microscope is a micron level. With UV light you can get to submicron level, but you will never be able to see 100 nanometers or less. But there are now optical techniques which you will get in there. It's called, where you can break the diffraction limit and look at ultra high resolution optical technique. Okay, you can look at that. But ordinary microscopes, you can see bacteria and these kinds of cells. Okay, so you can examine tissues, you can examine cells over here. So in our body, you have cells of different kinds. One cell, which has been really now very active area, is called the stem cells. And the stem cells are, you can say, virgin cells. They are not specialized in anything yet, and they differentiate. That means they specialize. Okay, and when they specialize, then they can form specific functions. Okay, so from stem cells you can get to skin cells, you can get to other kinds of cells, and then these cells, they bind together with in-between region consisting of some other chemicals called extracellular matrix. And when they combine together, they form certain kind of tissues. So you can have heart tissues or lung tissues or other kidney tissues. All these tissues are specialized to perform certain kind of functions, and they involve certain kinds of cells and certain kind of this binding glue that we call extracellular matrix. Okay, and then these tissues, they assemble together in certain shapes and functions, and that form organs. So you could have like heart, lung, and brain, kidney, liver. All these are organs, right? And organs together combine to form an organism like human. Okay, so very simple term. This is the hierarchy. So those of you who are chemists or physicists, maybe you will be involved at this level or this level, because here, as soon as you come here, you're looking at animals. Okay, whether in vivo or ex vivo sample extracted, and over here you're looking at a whole live animal. So these ones maybe initially would be intimidating to you to deal with this animal specimen. So you will do a lot of studies of your nanomaterials on these levels. Okay? All right. So that is like a human cell, eukaryotic cells, and you have different organs over here in organelles. So firstly, the cell is surrounded by a membrane, and membrane is these lipid structures, okay, mainly, and it's bilayer. We will look at it later, okay. And so the interior part is hydrophobic, and exterior part is hydrophilic, so that it can be dispersed in water, because you have a lot of body fluid is water, okay. You don't want to have something hydrophobic that isn't going to be stable in that environment, okay. And then inside you have these different compartments that do different functions. So nucleus is the largest organelle, okay, and it's the densest, and that is where the DNA actually replication occurs, and then RNA synthesis that migrates over here to do protein synthesis outside. Then other important organelle is mitochondria, where you have this energy factory, so it generates ATP and stores energy to release. Then there are Golgi apparatus, which is also another function, and so there are different compartments, different organelles do different things. So you may want to, most of the time that you're looking at diseases, you may be involved in nucleus and mitochondria, maybe Golgi apparatus, and then this whole area they call cytoplasm, okay? So then let's look at some of the bioprocesses that you may be looking at, whether trying to diagnose a disease or whether you are trying to just look at, understand the fundamental properties of cell or drug-cell interaction. You introduce some drug in there. You want to see what interaction is. One is cell replication. Cells replicate or reproduce and replenish the dying cells, process to reproduce and replenish. And there are two mechanisms. One is called mitosis, process by the nuclear divides into two genetically equivalent daughter cells. And the other one is called meiosis, specialized replication producing genetically non-equivalent cells, like germ cells. The other one is cell biosynthesis, production of cellular macromolecules, and this involves three steps. One is called transcription, where the DNA unravels, forms single strand, and then it attaches RNA to synthesize the messenger RNA, and so that one is called transcription. Then this one, actually messenger RNA gets into ribosome and produces protein. It synthesizes, connects amino acids to make the appropriate protein you need for a certain function. And the last one, which is called expression, this is activation and response of a particular kind of gene, whether it's internal or external. So firstly, you activate the gene, and then gene expression makes the appropriate RNA in producing the protein. So these functions are also affected by disease. So as a disease stays, you may want to actually look at all these three functions. Then the last one, again, I want to put is cell energy production. So cell to survive needs energy. So it is cellular machinery provided by two. One is glycosis, which is conversion of chemical energy to ATP, and the other one is APC synthesis. These are a lot of chemistry involved. Well, chemical energy is stored in certain bond, and when the bond hydrolyzes, it releases the energy for function. Chemical bond energy is in mitochondria through respiration, and light energy, or light is absorbed through chloroplast and stored energy that way. Okay? cell signaling, the cells that communicate with each other. That's a very important part. And these are the ones, hormones, that mediate communication between the cells. Okay, distant cells, adjacent cells, so on. Then last process is cell death. So if you're trying to kill a cancer cell or cancer, very often you will manipulate these. One of them is called apoptosis. So the cells, they have devised a scheme by which the disease cells automatically die. Okay, that process is called apoptosis. So anytime they recognize the cell is diseased, it commits suicide in a way, right? And that's what is apoptosis. And the other one is a cell killing through necrosis, where you can damage the cellular structure and then it dies. So what you want to do is, if you want to kill a cancer cell, then selectively induce these two processes or one of these two for that cancer cell to die. Okay? And the last is cell transformation, where in course of time a normal cell changes its genetic makeup and become a cancer cell, normal cell, because the DNA it mutates to is not really the regular or healthy one. Okay. Go to cells. In cells, as I said, you have cells, and then you have extracellular matrix consisting of molecules. They may be in the top of collagen fibers to give a structural body, and then they are connected, these cells are connected through these cell adhesion molecules so that they can communicate. And so when you are trying to look at tissue properties, it is not all just the cells. So if you study only cells, which is called in vitro studies, that only tells you a story of these parts. You know, when you are looking at a real biological system in your body, a tissue, you can manipulate these as well as ECM, extracellular matrix. So nowadays, the target is not just cells, but also ECM. So if a nanoparticle you want to have, you can embed activities here, or in membranes, or in extracellular matrix. Okay? And one thing, we'll come back to that later, is that our body has built in these barriers, walls, to prevent foreign things from getting inside and causing problems. Like a skin is a wall where certain size particles and certain size things cannot permeate through inside our body, but still oxygen and water can get through here. Okay? So that's skin barrier. So if you're trying to administer nanomedicine through skin, you need to tailor its characteristic so that it can get through the pores of the skin and get inside. That's called transdermal delivery. Similarly, for blood disease, our body has made this barrier, blood-brain barrier, that separates blood from getting inside the central nervous system. It lets the nutrients and a small molecule get in there that we need, but doesn't allow, the blood to get in there. Okay, so blood can carry and then get in there. So we'll look at how we can create nanoparticles that can cross the blood-brain barrier and get inside there to affect treatment for blood disease. Okay, let's see. So that is all the biology I'm going to talk to you about, and you can learn more about it. So again, the important things I emphasize: the cellular structure, dealing with the eukaryotic cell, what is the difference between virus, bacteria, and this, whether you can study each of them. And then the other one is cellular structure. Inside there, the organelles, and for certain disease, you may targeting your nanoparticle to one organelle, either mitochondria or nucleus or the membrane, because where it could be more effective to release the drug and so on. Then I talked about tissues, where I said that at tissue level, you can also target therapy to extracellular matrix, not just cell. Okay, And then we talked about the barriers, how you need to tailor, which we will come back to that later again. All right, so let me now say a little bit about physics. So the biologists can learn a little bit more physics. The chemistry and physics, the student, you already know about this. So light matter interaction, as I said, we use a lot of light-based diagnosis and therapy. So I thought I will review this part. And so this is a very fundamental concept that was developed by Einstein. First, these three. This is contribution of our Indian scientist, Indian science, Raman scattering. So absorption is a process where you have, as you know, atoms and molecules, whether it's in biological system or gas phase or in your laboratory scale, they have quantized energy levels, right? This is what quantum mechanics, quantum chemistry tells you. Energy levels are quantized so that you can only exchange certain amount of energy by absorption and emission. It's not continuous form of absorption. So here you have lower level quantized, higher level excited. A light can be absorbed to go to excited state. This absorption will occur only when light of this energy or frequency is incident corresponding to the gap. And that gives you absorption, so you can map out absorption spectrum, what frequency or wavelength is absorbing, right? That's absorption spectrum. The other one is emission. The light, when excited over here, many times it will be re-emitted, and that's what we call emission. And in microscopy, very often you will utilize the emitted light to map out the structure. That's what is called fluorescence microscopy, right? So we will talk about that. So the emission can be by two mechanisms. One is spontaneous. That means the light is spontaneously emitted in its lifetime of excitation, and then it decays, okay? So there are two characteristics. One is the emitted light energy or frequency or wavelength. The other one is lifetime of emission. How long will it take for this emission to decay fully? Einstein also proposed that you can have a stimulated emission. That means when you create a large population in excited state, it's called population inversion, and you bring in the photon of same energy as this gap, it triggers emission, and this is a stimulated emission, okay? And that's what formed the basis for lasing. The first is that you must have population inversion, and the second one is that you must have, by feedback, bouncing a photon of same energy coming to trigger it to stimulate. And that emission is not limited by this lifetime. And now C.V. Raman showed in 20s, late 20s or early 30s, I forgot, something like that, that actually there is another kind of transition. It's called inelastic scattering, where when the light goes as incident, it doesn't get totally absorbed, only it gets scattered to a lower energy, or in some cases higher energy. The difference between the energy of these two photons is what is from the medium or given to the medium. So when it is of longer wavelength or less energy, that means part of the energy has gone to the molecule to excite certain states. And in the case that he investigated, it was vibrational states. And then this light coming out is a lower energy. And that's what is called a Stokes shift, Raman scattering. So it became known as Raman scattering. So there you map out the difference between these two as a function of energy. And you see where this transition occurs, this photon, this difference occurs. Let's give you Raman spectrum, vibrational spectrum, as you know. And anyway, so these are the four things we commonly utilize in biophotonics. We look at absorption, like I gave you the oximetry, where light goes through and look at absorption spectrum, fluorescence, a lot of imaging we will look at later. You can also look at a stimulated emission in some cases to give you imaging. Then this Raman scattering is another one. You can do Raman imaging, or you can do chemical sensing using these Raman sensors. Okay, so we talk about those later. Okay, so this is a very important diagram that is very heavily utilized. You can utilize in biophotonics or healthcare. Okay, so when you excite, you know, in organic molecules particularly, let me describe that, is much more relevant. Organic molecules is that you have normally most of the organic molecules, they have closed shell structure. That means their spins are paired, right? Electron spins are paired, and that gives rise to what is called singlet state. The spins are paired, there is no net spin, there is no magnetism. Okay, so the ground state generally is S0, called singlet lowest energy state. There are some biradicals, radicals you can form where they are not the case, but we are not talking about that now. So when you excite light, the most probable transition is where they remain paired and go to higher level. Okay, and the higher levels, so they are also singlet, they absorb strongly, and you can have S1 is the first excited level, S2 is the second excited level, S3 is the even higher level. Okay, so you excite there, and normally in each electronic state you can also have vibrational levels. So you can excite coupled electronic and vibrational level. It's called vibronic level. Okay, so part of this energy is in form of electronic transition, part is vibrational. But what happens, the molecules, they very rapidly get rid of vibrational excess energy by what is called vibrational relaxation and come back to the bottom of the electronic level. Okay, and this vibrational relaxation is in sub-picosecond to picosecond, very fast. Okay, and that energy is converted into heat, but a small amount of heat. Now here there are choices. You can actually cross to another lower electronic level, S1, where now this pure electronic energy is converted into electronic energy for again some more vibrational energy. Okay, and this process is called internal conversion, because the spin doesn't change, stays the same, it's still paired. Okay, and so this one process is called internal conversion, same spin to same spin. Okay, and now from here you can either, and then part is again into heat a little bit, and then it has a choice. This is singlet and this is singlet, so spin allowed, it can emit light. That's called fluorescence. Fluorescence is emission of excited state of same spin as the lower state. There is no change in spin, so that's fluorescence. Or it can decide to cross to a triplet state, that may decide to change the spin and go to a lower state, which is generally lower than this, T1. In this process they have to change the spin, so it's a less probable process because it's spin forbidden, but under certain conditions you can do that. And then it can, so this is called inter-system crossing, ISC. It crosses from one spin to another, right? And then again you have electronic level and a lot of vibrational energy that you can get rid of, again by heat. Now this one is a long-lived level. It doesn't have, you need to change the spin to come down. So it stays longer over here, And this emission is long-lived. So in many systems, you can excite light, block it, and you can see the decay of emission. Okay, in seconds also. Okay, and that is because it's a triplet level. Okay, so this one is called phosphorescence. Okay, and you can have phosphorescence. Also, because it is a long-lived state, sometimes called metastable state. Metastable state, you can do photochemistry. It's more likely to do photochemistry from this. Okay, or you can do energy transfer to another system. And this is very important state for biology also. Then finally, the same intersystem crossing, rather than emitting light, it can cross to the ground state and convert all of them into vibrational excitation, and that one is heat. Okay? So look, you have many ways of getting rid of excitation energy, either through fluorescence, or through phosphorescence, or through photochemistry, or through heat. And you can utilize all of them into diagnosis and therapy. And you can design your probe, organic molecules or inorganic nanoparticles, or your nanomedicine thing, that you say, well, I want maximum fluorescence for imaging, fluorescence imaging. I don't want these competing processes to complicate. I want a photon get in, and most of the photons come out as an emission, bright emission. And you can design your nanomedicine probe that way, and we'll see that example. Okay? Or you say, well, I want to go through triplet state. Why? The triplet state is a good place, long-lived, to do photodynamic therapy, light-activated therapy. I want maximum population here. So how can we enhance this? That as soon as it comes here, it quickly goes into triplet. And we can design nanoparticles for that. Okay? Or you say, well, I want to do photothermal therapy. I want to use all the light absorbed into heating locally and fry the cancer cell, kill it. Right? Too hot, dies. Right? So I want this. So you can make very effective nano heater by manipulating that it doesn't go here, doesn't go here, it goes all the way here. Okay? The other thing is for imaging, photoacoustic imaging. Photoacoustic imaging also uses heat, and heat locally generates acoustic wave, ultrasonic wave, pressure wave, right? Because of the heat propagating. And that ultrasonic wave we detect by ultrasonic detection. So it's a hybrid of optical heating locally, right? Thermo-optic effect, and then ultrasonic for detecting image. Ultrasound, you do all the time ultrasound. You know, with the pregnancy, the baby, you go run ultrasound, right? Imaging. So that's photoacoustic imaging is a very important area. So this manipulation is very important. And you can manipulate through chemistry and physics, right? You need to understand the physics, and then you design your materials, synthesize it for maximizing one effect. Okay? And this diagram is called Jablonski diagram, after a Polish scientist, very famous. Okay, so now I want to talk about any questions so far? I didn't give you a chance. No? Clear or a little bit intimidated to ask a question? Yeah. Anytime. Okay? I want you to derive maximum benefit. I am giving my valuable time over here. Okay? Because I hope you'd benefit, right? Okay, so let's look at the other part. We look at the molecular response, how it interacts. So let's look at light itself a little bit. As you know, light is an electromagnetic wave, and it has electric field, and it has magnetic field, right? Most of the time we only look at electric field. When we're looking at interaction with molecules on, we look at electric field. That's why we talked about absorption, emission, scattering, and all those, right? So, and there is plenty of light sources in nature. Sun provides abundance of light, right? Also you have chemiluminescence, right? These organisms that glow, right? You have also, and these lights are incoherent as far as we know. We don't see lasing in nature, you know. Although we are already talking about some kind of lasing in space called random lasing that could be there, but as far as the light, sunlight coming here, or chemiluminescence, bioluminescence, all these are incoherent. They're going in all different directions. They are not truly monochromatic, even though they may have a color, you know, green color or other colors, but they still are broad. And so what they can do is only linear optics. And what do I mean by linear optics? How do I go back? Let's see if it works. Okay. Oh, no. Yeah. Linear optics means one photon doing the job for one molecule. So you have one photon being absorbed, or one photon being emitted, one photon being scattered to another photon. This is what is called linear process. Okay? Why? Because the light is not as strong, it's not coherent, so you do linear one photon interaction. But what we made, this is an old slide, so excuse me, now I have changed it as a human-made, not man-made. Okay? So human-made light is coherent, because we have lasers. It could be very intense. We can make it in a very short pulse. Now we are talking about attosecond pulses. Okay, so it's 10 minus 18 something. Okay, very short pulses, and they are so intense that they can drive nonlinear optical effects. That means interaction of more than one photon together with a molecule, right? And that's what is called nonlinear optics, and that involves multi-photon interaction. So let me give you an example of those. And these ones we utilize also for nanomedicine, as you will see later. So this is a little bit of physics here. At least we have one physics student, right? Okay. So what happens when light is incident on a medium, whether it is your molecule, or whether it is a solution, or whether it is a solid phase or polymer bulk film, you involve interaction of electric field of light with the medium. And the medium has electron, a nucleus. So in that electric field, which is oscillating like electric field, right, it polarizes and tries to induce dipole moment. That means separation of electron from the nucleus, push it further, okay? And that's called dipole moment, right? So what happens in the medium, the dipole moment, they all give rise to, together, collectively, a polarization of the medium. Is polarized in certain ways, electron cloud, and that polarization in ordinary light is only dependent on electric field strength, E. This one is called susceptibility of the medium. And the real part of the susceptibility actually gives rise to refraction, scattering, diffraction, and all that. The imaginary part of that gives rise to one photon absorption. Okay? But when you have intense field, like laser field, very short, very intense pulse, the electric field is very high. So you cannot just stop at this level, you have to use a higher order term. And these are the higher order terms that give rise to nonlinear effects. multi-photon processes. So this one, for example, is what is called Chi2, is quadratically dependent on the electric field. So electric field is weak, like an ordinary incoherent light. These things don't affect. But intense field, it occurs. But this one is not so important for many of the systems that you deal with which are disordered. This gives rise to second harmonic, and it requires that the medium be not symmetric. But when you look at biological system or plastic, so on, it's random. It's non-centrosymmetric, so this doesn't happen. Then you look at this, Chi3, third order effect, that one gives rise to 2-photon absorption. So 2 photons being absorbed, going to excited state, then it comes by internal conversion to lower state, which can emit light. Now look at this one versus single photon absorption. Single photon absorption will, the absorbed photon is of higher energy than emitted photon, right? Part of it goes as a heat, right? But this 2-photon absorption, the absorbed photons, if they are the same energy, most of the time what you would do is the same energy, you will need only half of this gap for each photon. It needs to kind of only half of energy. So suppose this gap corresponds to 500 nanometers in the green region, you need these ones only 1000 nanometer photons in the IR. But 2 of them get immediately grabbed and go through higher level. There is no level here, energy level, it's called virtual level. And you just combine 2 of them together, get here, then you emit light. Now look at this light. This light is of higher energy than this, each photon. So this process is up conversion. The emitted light is of higher energy than absorbed light. Okay? Similarly, the 5th order term can give you 3-photon absorption. 3 photons can be absorbed and emitted. Okay? And this is again, remember I said in my opening remark that I want you all to be imagining big and thinking unthinkable outside of the box. This thing came out of imagination of a Polish scientist, Maria Gopotmeyer, in late 20s. And she theoretically proposed that under certain intense condition, a molecule, an absorber, can absorb 2 photons simultaneously, not through real level. It's not climbing up here and here. It takes 2 photons together and goes up there, right? And she proposed theoretically that there is a 2-photon absorption process. Weak in most systems, but it should exist. And people say, oh, okay, the calculations, yeah, it could, but what good is it going to do? It's very weak, it will never be practical, right? And then with lasers and short pulse lasers, it became reality, okay? And in 1990s, we made, again, further advancement came through materials chemistry, organic chemistry, and physics. We try to understand what kind of structures can give a very large 2-photon cross-section and absorption. And we made those materials, and we showed that with very low intensity pulse lasers, you can do very efficient up-converted emission that you can utilize for bioimaging. And now 2-photon microscopy is utilized worldwide. Most biological labs in America have 2-photon microscope. So something that was very fundamental, very theoretical, perceived to be of no importance, is now given rise to what is called 2-photon technology. A 2-photon technology is not just for imaging, but you can utilize this absorption of subsequent to the triplet state or someplace where you could do photochemistry and you can do photopolymerization. And that polymerization is very localized because this process is most effective where the intensity is high. And where is the intensity high? When you take a laser beam or light beam and you focus, at the focus point the intensity is the highest. That's where most of the emission will come out, or that's where most of the photochemistry will come out. So you can do micro nano polymerization and fabricate three-dimensional object by varying the focal point and scanning it and making it your objects. Okay, so people have made 15 nanometer polymeric discs or objects, and others by using two-photon polymerization. So this is a very important nonlinear optical effect that we will see. 3-photon is less because you need even a stronger intensity to do this effect. It's a higher order effect, more dependent on the field. Okay. So I'm going to talk about upconversion, and you will see later in examples of upconversion that we are going to utilize. So what are we talking about upconversion? You have a medium, could be biological medium, or the material you have synthesized, organic molecules or inorganic nanoparticles and others. You are sending a longer wavelength light. And remember, I said the emitted light coming out is a shorter wavelength. And so it's upconversion, going into higher energy. Okay? And as I showed to you, multi-photon process, 2-photon is one example where you can actually go through this third order effect, 2 photons being absorbed simultaneously and getting emission. Okay? The other one is through second order effect that I told you requires non-symmetric structure. And you have green pointers. You see green pointers. In green pointers, you have a light that's generated by lasing is 1.06 micron, 1064 nanometers. And then it goes through a material that doubles it to 532 color, and 532 is green. So your output is coming out green. You use it, small handheld laser pointer, these effects. But these upconversion of lower energy to higher energy occur through virtual level. There is no energy level. It's not like it goes from here to here, here to here, or here to here, here to here. One step process. But so that's called virtual state. These they're intensity driven. You need an intense electric field of the laser light. Then there is another kind of upconversion process that we shall see also applied to biology and nanomedicine, where you actually have a lot of levels close by. Okay? One example is rare earth ions, where you have F levels. Rare earth ions, they have sp, df, f orbitals. There are lots of F levels, and F to F transitions. Many F to F transitions are low level, so that you can take one photon to go to first level, and then another photon from there to another level. And you can climb up like a ladder. So there is a step here. Here there is no step. So these are linear transitions, but still they are quadratically dependent, because if 2 photons are absorbed, and they can then emit from here, which is again upconversion. But this upconversion is through real state, real level. And that means you can do it, you don't require intense laser pulse. And we can do this thing, upconversion, through lamp or very low power laser, continuous wave laser. No expensive laser is required here. So that's the beauty of it. And there are two. This is what you call excited state absorption. But in rare earth ions, they also occur another process called energy transfer upconversion. So the two ions together each absorb a photon, but if they are neighboring close by distance, one ion gives up its energy to excite the other ion to upper level and returns to lower level. This is called energy transfer upconversion, because now this second ion is excited here and it can emit photon of lower energy, higher energy. Okay, so this is upconversion through energy transfer, is upconversion to excited state absorption involving real states. And the beauty of this is that we can use a low power light source to do upconversion. Okay, is there 1 minute. Sometimes it works, sometimes it doesn't. I want to go back. No, it goes forward. Yeah. Okay. So is this difference clear to all of you, students? The difference between this and this? That this is through a virtual state. There is no intermediate state actually real. And it is one-step process. And this is two-step process. This requires different structures. Organic molecules generally, you have this, okay? Because organic molecules generally, these gaps, intermediate gaps, are not there appropriately positioned to do this. Rare earth ions, a lot of inorganic materials, you can have this kind of thing. That's why there is also an emphasis from the chemist perspective marrying organic and inorganic together, okay, to make hybrid kind of materials, you know, and we will talk about some of those. Okay. Or if you don't have any question, let's move on. So again, going back to this, as I said, 1931, Maria Gopper-Mayer published first paper, the laser with laser, first demonstration of two-photon absorption was 1961. So there is imagination to reality, and then leading to a technology called two-photon technology. In 1990s, we showed two-photon microscopy with our new materials. We design and synthesize very high two-photon absorption, upconversion lasing at the upconverted output, two-photon data storage, two-photon microfabrication, and two-photon photodynamic therapy, killing the cancer using light. Okay. So, and this is, as I said, a case of transition through the photon upconversion through real state, rare earth ions. And that we like to call, we implemented it in a nanoparticle formulation. And we call those nanoparticles nanotransformers. They're optical nanotransformers, so they convert light from one color to another. Okay? And so we synthesize them, a very simple colloidal chemistry, and you will see the examples of that later, how we make it, a materials chemistry. So inorganic chemistry generally is much simpler than many-step organic synthesis. Right? So we make them, and these nanoparticles generally are anywhere from 5 nanometers to less than 100 nanometers, depending on what we want to do with them. Okay, and these nanoparticles then have coating on the surface. That's what's called surface chemistry, with ligands or surfactants, which make them dispersible in solvents so that they don't aggregate together. They're held separately. Okay, and for water, we want to have surfactant. That outer one is hydrophilic, so that they can disperse in water, right? So here is those nanoparticles dispersed in this vial, very transparent, and you can't see anything going through them, right? So the letters are very clear through there, very transparent. And then since they are nano, they don't scatter light. They're not absorbing visible light, so you don't see any color. Then when we shine light at 980 nanometer with a handheld even laser light, like this kind of laser size, you can do, depending on the rare earth ion present in nanoparticle, you can have up conversion to green. So there is invisible light going through, 980 nanometer you cannot see. You can convert to green. If it has erbium ion, we generally use fluoride lattice. Okay, so erbium ion, it will give you green or red color up conversion, as you see this streak. So as soon as you shine light through here, you will see green, red streak, or another formulation green streak, or another formulation blue, if it is thulium. So 980 IR to blue color. Actually, you can convert all the way into UV by not 2 photons, but it can climb up by 1, another, 3, 4 photon climb up also. Okay. There is another process we are investigating also, that is called quantum cutting, where it's a reverse process. You can take a photon of high energy, maybe in the blue, maybe UV, and then it gets absorbed here, and then it emits 1 photon to this level, and then another photon back there. So you have 2 photons of lower energy being emitted, that we call quantum cutting. Okay, and there are many different applications. Primarily we are looking at, in this case, converting invisible IR light for night vision to visible. So that's engineering application, photomedicine, and solar energy conversion. So harvesting IR photon absorbed, emits in the visible region to add value to solar cell. This one is display, so a high energy UV photon can be split into visible photons to enhance brightness. So we are looking at these applications. Of course, we will talk only about this photomedicine application. And we call it on-demand, on-site photon conversion. And we'll see what I mean. I may find it easier for light of one wavelength to go deep inside a medium like biological medium, and you will say, for example, infrared light can go deep inside, but when it goes to disease site, that infrared light may not be active. It doesn't have enough energy to do any activation of drug or therapy or something, right, photodynamic therapy. But if you can convert with a nanoparticle sitting there into a green light or blue light that you need to activate a therapy or activate some biological process, stimulate neurons, then it can be useful. So that's what I'm saying, that you are on site and on demand, you convert color, one color into another photon that you need to activate a process. So we call on-site, on-demand. Okay. We also look at nonlinear effects, which are other kinds where there are 2 different photons incident of 2 different wavelengths, and the difference between that goes to excited vibrational mode. And this spectroscopy is called coherent anti-Stokes-Raman scattering, because it's generating vibrational excitation but nonlinear interaction of 2 different photons. Okay, and we utilize that for imaging, vibrational imaging. Then we can have 2 photons interact together, add, and give you a photon of higher energy. We call some frequency generation we use for imaging. And these 2 I already talked about, upconversion, emission. So we combine all these together in our biological investigation. Now the other thing that one can utilize is not just intensity of light emitted. We stop at 1 o'clock? Or okay. So let me finish a couple of slides, and because we spend a lot of time in general things, but don't worry, you know. So here, and you all have the notes, right? The lecture notes, they have all of them? Okay. So one thing I would advise, you read again, and in the next lecture, in the beginning, we will take 10 minutes for you to ask any question that by reading your notes and or rethinking, you may want to clarify. Okay? All right. So also, you can utilize time resolve experiments to get more information about time changes. And one other thing in the biology is the biological processes occur in very different time scales, different processes. Like in your eye, retinal changes as a process of vision, some of the initial changes can be in hundreds of femtoseconds. And then you're looking at cell replication processes, it can be hours, right? Disease manifestation or disease progression can take months and years, right? So there are biology involves different time scales. And what light allows you to probe all these events and all these multiple time scale. That's the beauty of it. You can do ultra fast processes because you have pulses now, very easily femtosecond pulses available, and so you can look at these changes in eye, retinal, how it transforms initially to create electrical impulse. You can diagnose that, or you can profile. So here is an example of this that we utilize. One of them is fluorescence lifetime imaging. So the microenvironment is very dependent on what kind of tumor you have, right, or cell you have. Cancer cell has very different microenvironment than normal cell. So there, if you're looking at fluorescence, you have embedded some material, your nanoparticle that has fluorescence, or you're looking at intrinsic fluorescence of NADH or some biological component. The lifetime decay of fluorescence is dependent on its microenvironment. So by looking at lifetime and mapping the distribution of lifetime, I can also use it as a diagnostic tool to see whether the cell is normal or it is partly changed, or the disease progression is further produced, right? So lifetime is very important parameter, and you can map out in microscope lifetime distribution. That's called lifetime imaging. Okay, so you can buy commercial system. We have a number of them in our lab where we can do lifetime imaging. The other one is fluorescence recovery after photobleaching. Organic molecules, when you optically excite for a long time, go through cycle of absorption emission, they bleach. And most bleaching is chemical, they alter, okay? And so what happens is that if you have a protein that actually is either emitting itself, like green fluorescent protein, or an organic dye that is attached to that protein, what will happen is that if you focus intense light in one region, it bleaches. And there is no emission from there anymore. So you have bleached out this dark spot. And then you remove that intense pulse or reduce its intensity. Then what happens is the medium is not stationary. Things are moving around the cell. Cell is dynamic. It's not fixed unless you fix it. Paraffin, use that and immobilize in the glass slide. Things are moving around. And if you are moving around, that region that you have bleached out will get out of the observation region. Another fresh one will come in there, and you will see the intensity of fluorescence recover, right? As they move in, the intensity goes up, right? So fluorescence recovery after bleaching. And what does it tell you? It tells you the mobility inside the cell. And that is different for cancer cell than a normal cell. So you can use this method, and biologists, they like to use acronyms. So this is called FLIM, Fluorescence Lifetime Imaging. This is called FRAP, which is Fluorescence Recovery after Photobleaching, that talks about the dynamic environment of a cell. Then the other one over here is called Fluorescence Resonance Energy Transfer Imaging. So you're looking at protein-protein interactions. That may be one mechanism for cell communication or cell function. The proteins, two different proteins are interacting together, and you want to map out how these interactions manifest. Okay, so then you can tag one protein with one molecule, it's called donor, at a higher energy level, another protein with an acceptor. This is an organic probe of lower energy. So when you excite this, and if these two proteins are close enough to interact, and you excite this donor, is of higher energy, it gives it to the acceptor, and you have acceptor fluorescence. Okay, so as long as these two proteins are interacting close by, you always see emission from the acceptor molecule, lower energy emission, when you excite the higher energy donor. But if they are not interacting, they are not close by, then when you excite this, you see emission only from here, not from here. So by looking at fluorescence energy transfer imaging, you can map out the domains where the two proteins are close together and interacting. And that's what is called fluorescence. Actually, it's called fluorescence resonance energy transfer. But the physicist who invented that energy transfer process, it's called Forster, was the one. So physicists like to call it Forster resonance energy transfer, but acronym in each case is FRET, whether you call it Forster or fluorescence. Okay, so this is also a powerful tool, imaging tool, to map out the areas where you see the protein interaction of certain kind. So these are all time resolved studies that, in addition to intensity distribution fluorescence imaging, one can use. So fluorescence energy transfer imaging has certain requirements. Okay, and this is where the physics comes in. You need to understand the physics to design two different molecules that can interact together to give energy transfer, or two inclusions in a nanoparticle. First of all, they need to be within nanoscopic distance, a very strong dependence on R, the energy transfer. If the two are very far, they don't. So that's called this FRET radius, and for most organic molecules the radius is less than 10 nanometers. Okay, so this is why nano formulation is very important if you want to use FRET for sensing or others, because if you make 10 nanometer nanoparticles and you put these two dyes in there, or proteins, whatever, they are forced to interact. They are within nanoscopic distance automatically. The second thing, a requirement, this is expression, I don't want to go into details, other factors, but this is the distance dependence, and depends on the dipole strength and other factors. And the other one is a spectral overlap. So the donor absorption fluorescence, donor fluorescence should overlap with absorption band of the acceptor. If there is no overlap, there will be no energy transfer. Okay? And there is a larger overlap, so the overlap integral between this curve and that curve gives you the efficiency of energy transfer also. So you want to match. So when you have two tags, look at protein interaction or embed energy transfer, you need to maximize this overlap. Okay? This we already talked about, how we can bleach and map out the mobility inside a certain region. Okay, so shall we stop here? And then, huh? Okay, so I will now talk about different imaging modalities we use in biomedical imaging. First, and then we'll talk about optical imaging in a little more detail, give you some examples of that, and move on. Okay? Any question so far? Yes. Part of what?
Part of the software.
Partly it is software, and partly is your materials design, right, that you use in there to probe. So, you know, you need to have software, and you need to have appropriate hardware also, where you're looking at how you're going to bring an optical train to bleach it, and then to monitor the recovery, emission. So those things, the software package, when you buy a microscope, you could get software package from them to all these film, fret, all these you can get from them. From the microscope company, you can get that software. And they also will provide you with the light source, detection system, gating, all those can be provided by them too. And depending on if you're doing, see, let me give you, when we come to microscope, we can talk about it more. The tools, the cost, you can ask. These are not cheap. The only cheap microscope is epifluorescence microscope, where you can just kind of look at fluorescence mapping in 2 dimensions, just plain. Okay? Another question? Yeah? Multi-resistant, yes.
Suppose the disease which is cross-reacting, like TB as well as HIV, and leprosy as well as HIV. In this case, if you want to design a new drug with the help of nanotechnology, how far you can get a success?
Well, actually, we published a paper, for example, tuberculosis, some years ago. The important thing is that you need what nanoparticle provides you is that you can use multimodal therapy, and we'll give you an example of that. You can include different kinds of drugs, because a nanoparticle doesn't have to be simple nanoparticle. It could be hierarchically built, different domains, and different domains can put different things. You can also put what is called gene therapy, so modulate immunoresponse. We'll talk about that when we come to nanomedicine therapy. So there is some partial success, still long way as a promise. Okay? And you had a question. You're saying about the faster energy transfer, fluorescence energy transfer? Your question is no. Can you come here, because I can't hear you very well? You should have a microscope. Yeah. Don't be afraid. It's okay. Yeah. Fluorescence, yes. Okay, very good point. Actually, we have a paper that says phosphorescence imaging. Okay, phosphorescence bioimaging. The problem is this: there are twofold. One of them is a longer lifetime. So fluorescence is in nanoseconds generally. So you can scan fast, and you can acquire images very fast. Okay. Phosphorescence, the best you could do is in microseconds. Okay. And so it's much slower process. That means your data acquisition is going to be a little bit slower. However, you can take advantage of another, it's called time gating. And time gating, you can reject unwanted signal which are very fast. So you don't collect for 100 nanoseconds, you don't collect any signal, and correct after that, so you only see phosphorescence. That has advantage. So one limitation is data acquisition. Okay. The other one is that the nature of the triplet, which works for photodynamic therapy, but it hurts in other cases, is that phosphorescence is very sensitive to oxygen, because the triplet state likes to transfer energy to oxygen, triplet oxygen, and which is good for photodynamic therapy. But if you are trying to collect image by phosphorescence and there is oxygen, it will invariably quench the phosphorescence. But fluorescence is not as sensitive to oxygen. Okay, so for those reasons, phosphorescence has not been utilized very extensively, but more and more people are realizing that this triplet state is very important also. And one example is not just for bio, which we showed that some of these porphyrins, we can have very good phosphorescence and we can use imaging and we can advantage of long lifetime. You can look up that paper, okay? But also in LED, or getting light emitting diodes, which originally injecting charge and looking at fluorescence, okay? But now there is also, they call phosphorescence LED, where you can also use phosphorescence emission for light emitting diode. So there are a number of companies now working on phosphorescence LED. So phosphorescence has merits, have not been fully explored, and the problem has been these two, lifetime and oxygen dependence. Okay? Any other question? No? All right, then go and think, right? And we will meet again. I will see you tomorrow, and you have a hopefully successful afternoon. And again, look, the reason I used to distribute cards, but it's no sense. You can find everything about me in the internet. Go on Google. You can find everything. I cannot hide anything, you know. I mean, people, a friend of mine found out that I had given some lecture in China, someplace, and I didn't inform him that I was coming to China, and goes in Google and sees, and right, how come you came here? I didn't know anything about it, right? So you cannot hide anything on the internet. So you can find my email easily and just write to me if you have any question, and I'd love to hear, because that's the best reward, no matter what I get and what I, compliments I can get. The best compliment is from a student writing to me that I really like this thing, and I learned this thing, or I want to know this thing, can you send me this? I'm very excited about it. And that's the kind of reward I'm looking for. I'm looking for reward that it does benefit you in some way, inspires you, make you think some new ideas, and don't think this is a crazy idea. Maybe it is, but that's a starting point, thinking outside of the box. Okay? All right, thank you.